So I think we'll start. Good afternoon, good morning, and good evening, colleagues. Welcome to today's IUIS webinar. We have 85 attendees on the line. My name is Wendy Burgers. I'm a viral immunologist working at the interface of HIV and TB immunology, although now I spend all my time studying immune responses to SARS-CoV-2, and I'm at the University of Cape Town in South Africa. We're not talking about infectious disease immunology today, but something equally as exciting. We all know about the revolution that's taken place in cancer treatment in the last years, and immunotherapy has really changed how cancer is treated. But the response rates still remain low, in part due to resistance mechanisms in the tumor microenvironment. So in today's seminar, Greg Delgoffe will discuss how the metabolic features of the tumor microenvironment can shape T-cell differentiation to alternative fates. And he'll also highlight how metabolic reprogramming can be enhanced to promote improved anti-tumor immunity. Greg's an associate professor of immunology at the University of Pittsburgh. He directs the Tumor Microenvironment Center at the UPMC Hillman Cancer Center. He obtained his PhD in 2010 at Johns Hopkins and did a postdoc at St. Jude's Children's Hospital. He's had his lab since 2014, and they've been working to understand the metabolic deficiencies experienced by T cells that infiltrate tumors and leverage those insights to bolster anti-tumor immunity. His group has shown that metabolic defects are central to T cell dysfunction in cancer, suggesting that immune-based therapy can be improved by modulating the metabolic state. So today's seminar will be about 45 to 50 minutes, after which we'll have a Q&A session for about 15 minutes. Do type your questions in the Q&A box. You can make comments in the comments box, but we'll be picking up the questions from the Q&A. So put your questions there. And after the talk, I'll be asking Greg some of your questions. Today's webinar is supported by Thermo Fisher Scientific. Thank you to them, and welcome, Greg. Over to you.
Thank you, Wendy, for the introduction. That's very kind, and thank you for inviting me. I'm very excited to be talking to this esteemed group of individuals. I will go ahead and share my screen here, and just make sure that I can get my pointer up and running. So today we're going to be talking about metabolism and immunity, but really in the context of cancer and the kinds of environments that it can produce. So today this is the kind of the broad kind of title, but really today it's a tale of three environments. It's kind of a play on some words, but it's stuff that we're very excited about, and I hope to get you guys excited about it too. These are my disclosures, none of which are actually completely relevant for this talk, except for one slide, in which there's a relationship that I have with Bluebird Bio, which manufactures CAR T cells. So as beautifully highlighted by Wendy, immunotherapy has really changed the treatment paradigm in cancer. And when we talk about immunotherapy, it's not just what you read about in the New York Times. There's actually many different types of immunotherapy, and this is from a review article that we talked about at least 3 classes of immunotherapy that are currently FDA approved. So there are therapies like cellular therapies, in which CAR T cells or expanded tumor infiltrating lymphocytes can kind of numerically augment a patient's anti-tumor immune response simply by growing them in vitro. We have an underappreciated form of immunotherapy that's FDA approved called oncolytic viruses, which can vaccinate a patient against their own tumor, but also inflame the tumor microenvironment. But most of the excitement has been around the blockade of inhibitory receptors like CTLA4 and PD-1. Also new to the game is LAG3, to kind of reactivate a patient's dormant anti-tumor immunity in situ. And this, of course, has received FDA approval in many indications that I kind of struggled to keep up with, including many that are in the first line, which I think is a very exciting thing for cancer therapy, that people are being treated with their immune system first. And I think that's really exciting. Now, the reason why immunotherapy is so exciting is that the responses to immunotherapy can be rapid, but more importantly, they can be durable. And that's what you're really looking for with cancer treatment, right? Is our therapies that last for a very long time. But of course, what we know is that, and it's kind of the $1 trillion question in the field, is only a subset of patients receive that benefit. It's this tail on the curve. And this 10% is a very significant 10%, but it's one that's, for instance, with CTLA-4 blockade, it's what got CTLA-4 approved. But the fact is, why is it only 10%? Why does immunotherapy only help some patients? And I think the big key question, question correlative to that is, how is immunity regulated at the tumor site? Because that is where the immune system really needs to function, is within the tumor bed. And that's kind of where my laboratory comes in. We try to understand this tumor microenvironment, this tumor site where the immune system is functioning. This is a place that we need to be thinking about. So we've known for several decades that this tumor site, the tumor microenvironment, it engages in some very immunosuppressive functions by its very nature, and there's a few listed here that we know all about, because the tumor is not just a bag of cancer cells. It's normal, tumor, and immune that are all together. Now, but a common phenotype of cancer that we've known about for a few hundred years is that it's hungry, right? Is that cancer cells, they need to continuously divide, and to do so, they need to become metabolically deregulated. And so we've known this for a very long time. This is a section of a mouse tumor, which we've infused the animal with a chemical tracer for hypoxia called pimenidazole that we can stain for with an antibody. That's the red. And we've also infused this animal with a glucose tracer to see what cells take up glucose, akin to FDG that's used in PET imaging. And so you can see that it's a very heterogeneous environment with some areas of extreme metabolic activity and some where it's just hypoxic and some where there's just glucose uptake. It's very interesting. So this tumor microenvironment that's driven by the metabolic derangement of the tumor cells themselves creates this landscape that's distinct amongst tissues, creating both a sink for essential nutrients as well as a buildup of potentially toxic byproducts. Here are two examples. And this is important because this might be a patient-specific variable, that some patients may be hungrier than others in terms of their tumor. And so we kind of decided to ask this a few years ago, where we really started to explore melanoma patients, which is, of course, the poster child for immunotherapy currently. But we analyzed the metabolism of the tumor cells directly from patient biopsies prior to initiation of anti-PD-1, and we put them into a seahorse instrument, which allows us to measure the metabolic flux of those cells in real time. So we can measure the oxygen consumption of the tumor, as well as the glucose consumption of the tumor. Again, from these, for each one of these dots represents a patient. And I hope what you can appreciate is that there's extreme heterogeneity in patients that had not begun to receive immunotherapy yet. And we characterize the microenvironment of each of these individuals, but to make a very long story short, if we look at who went on to progress on anti-PD-1, it's these patients that have these hypermetabolic tumors, especially those that deregulate their oxygen metabolism. So if the tumor cell consumes oxygen, those cells, those patients are more likely to fail immunotherapy. And you can see that here, that with a high OCR, you can split the response to anti-PD-1 very significantly. So anyway, long story short, why is that important? Well, when you're trying to ignite an anti-tumor immune response, well, this is extremely energetically demanding. That immune activation and function is a really, really big deal. So, this is something we've known about for the last 20 years or so, and has really been appreciated in immunology, is that T cells, unlike maybe, kind of your typical fibroblast or something like that, they need to exist in extremely quiescent states, in naivete and in memory, essentially persisting for a lifetime without really dividing. But upon activation, they need to engage in extremely rapid cell cycles. They need to proliferate. They need to be biosynthetic, and ultimately need to be able to infiltrate and kill target cells. And so they rapidly change their metabolism in order to meet their energetic and synthetic demands. What you can see here, this is a measure of glucose metabolism, specifically the capacity of cells to ferment glucose into lactic acid. It's the reason why your media turns yellow when you culture cells. But if you activate T cells, within minutes of activation, those cells dramatically change how they perform glucose metabolism, and it stays that way throughout their differentiation. And over the course of activation, they begin to change the way that they take up glucose, and they start to become very competitive for a glucose tracer. And as part of that, they also be able to conscript kind of generally appreciated nutrient sensing pathways, AMP kinase, HIF1 alpha, mTOR, to kind of guide the differentiation decisions that T cells need to make after activation, becoming inflammatory or suppressive, memory-like, or exhausted. Now, long-lived cells. Now, all long-lived cells are characterized by this, but in T cells, we call them memory T cells. Long-lived cells are characterized by having a high mitochondrial mass and this value that I'll continue to talk about during the talk, called respiratory capacity, or spare respiratory capacity, which is the ability to engage oxidative metabolism in stress situations. And so this value that we get, this uncoupled oxygen consumption with the difference between this basal rate and this maximal rate, this is actually very indicative of longevity, of cells that live for a very long time. T cells, fibroblasts, you pick it. So we asked some questions about how do T cells behave metabolically as they enter various environments. This is something that has vexed me since, really since I began training. Like, how do T cells interpret their environments? And so these are the three environments we're going to be talking about today. First, the capacity of tumor cells to consume oxygen and produce hypoxia, the coincident production of ROS that occurs during that. And how that changes cytotoxic T cells into becoming more dysfunctional, to change their differentiation into a more hypofunctional fate. We're also going to talk in the second part of the talk about how tumor cells deregulate their glucose metabolism, and they produce a lot of lactic acid, and how this can change the differentiation of CD4 T cells into more regulatory fates. And then finally, I'm going to finish talking about a third type of environment that is really important in certain kinds of cancer immunotherapy, and that's the in vitro environment in which we grow therapeutic T cells, like CAR T cells, and how this may prime them to be short-lived in vivo. All right, so I'm gonna take a step back to a few years ago. This is the first paper that really came from my group, where we really started looking at T cells that infiltrated tumors, but from a metabolic lens rather than like a phenotypic lens. And what we found, using a tracer for glucose that allowed us to track glucose uptake, and a dye that stains functional mitochondrial membranes, we looked at T cells that were infiltrating experimental melanomas. This is B16. And what we found is that when we looked at CD8 T cells that infiltrated tumors, they had some very, very striking metabolic defects, specifically a repressed ability to take up the glucose tracer, but a really striking loss of functional mitochondria. And we confirmed that loss of functional mitochondria in using electron microscopy. You can see these are lymph node resident T cells that have kind of textbook mitochondria. These are the mitochondria you see in tumor infiltrating T cells. They're small, they're malformed, they don't possess a lot of structure that allows them to do chemistry. Now, this occurs across solid cancers. We found this in mice as well as in human patients. And to sum up a lot of data, the degree by which, the severity, if you will, of the loss of mitochondria was actually variable in the patient population, and it correlated with the response to PD-1 blockade. So if you had more mitochondria in your TIL, you were more likely to respond to anti-PD-1. What's more important here is that it's not that T cells entered into tumors and they dumped their mitochondria, but rather it occurred progressively as T cells differentiate to this kind of alternative fate that we call exhaustion. So as they differentiate to terminal exhaustion, kind of they begin to upregulate PD-1 and ultimately turn on multiple co-inhibitory receptors, the canonical one being TIM3, is that they lost mitochondria over time. and that it occurred due to the downregulation of this molecule called PGC1 alpha. And PGC1 alpha is a transcriptional coactivator that programs this process that we call mitochondrial biogenesis, which is the generation of new mitochondria. And so you can see, if we looked at PGC1 alpha expression by protein, you can see that this molecule gets repressed as T cells differentiate. So now these things could just be coordinates. And so, could you improve the mitochondria and ultimately improve T cell function? So to do this, we took antigen specific T cells. These are just OVA specific T cells, and we simply overexpressed PGC1 alpha. And when we do this, we actually endow those T cells, this is just in vitro, with that respiratory capacity value that I talked to you about before, this kind of feature of memory T cells. And just by overexpressing this kind of mitochondrial gene. And so then we used those T cells and transferred them into mice that were bearing small OVA-expressing tumors. And so the tumor expresses the antigen, of course, and so the T cells can go in. And so when we see those T cells enter, you can see that if we overexpress PGC1 alpha, pardon me, I'm just getting over a cold. Those T cells are resistant to the loss of mitochondrial mass that's normally experienced by their empty vector brethren. And you can see that tabulated here. But far more importantly, those T cells are more functional. They can produce more cytokines per cell. And I think far more importantly is those T cells can actually mediate tumor clearances in a significantly more larger fraction of mice, and all mice receive benefit. So at the time when we were doing this experiment, we thought this was pretty cool, but we were really just doing adoptive transfer, just to kind of see what the features of these T cells really are when they enter the tumor. But it wasn't until now that we realized that we were really doing cell therapy. And so we kind of asked some questions about whether or not this could translate into CAR T cells, because CAR T cells, of course, we have to genetically modify to kind of redirect them towards tumor antigens. And so could we apply the same logic to CAR T cells? Well, before we could do that, we had to kind of confirm that CAR T cells also succumbed to the same kind of metabolic exhaustion that murine TCR transgenic T cells did. So to do this, we utilized human PBMC, and we transduced them with a vector that encoded a CAR based off of cetuximab. So cetuximab, of course, targets the EGFR. And so these T cells can kill EGFR high tumors. Notably, this contains a 4-1BB co-stimulatory domain. And we use them to treat immunodeficient animals that were bearing A549 tumors, which express lots of EGFR. And notably, these CAR T cells are not functional in this state. Like most CAR T cells, we haven't had success yet in solid tumors. So we asked, do these CAR T cells still succumb? And the answer is yes. These CAR T cells, if you get them from the tumor microenvironment, they respond very, very poorly to their cognate antigen compared to the ones that are in the periphery. But far more importantly, those T cells also lose mitochondrial mass. So we now could suggest that maybe we could improve this. So could we actually, in the interest of time, I'm actually going to skip over this and actually just go to the gusto. I don't want to run out of time today. But you can see here that if we overexpress this kind of, we have an engineered form of PGC1 alpha, you can see that we can again endow those human CAR T cells with that respiratory capacity value that I alluded to earlier. And we took these cells and we treated mice that were bearing the A549 tumors. Now, remember what I told you at the beginning, the wild type CAR T cells don't work very well in this situation, and that's absolutely the case. When you give CAR T cells, there's a little bit of tumor aggression, but really no mice ever receive any major benefit. But if the mice receive PGC1 alpha reprogrammed CAR T cells, those CAR T cells work phenomenally well and mediate clearances in a number of mice. And more importantly, when you re-stimulate them and isolate them from the tumor, you can see that they function. They can function more effectively. So this is a single metabolic gene. One metabolic gene was enough to metabolically reprogram therapeutic T cells, prevent functional exhaustion, and ultimately improve anti-tumor immune function. But this is really important. What does that mean when we talk about exhaustion? And exhaustion is a term that gets thrown around a lot in cancer immunology, but it actually is a defined feature of T cell differentiation. It was originally described, of course, in chronic viral infection by Rafi Ahmed and Ralph Zinkernagel, many others. But exhaustion is really a type of hyporesponsiveness that arises from chronic stimulation, from persistent antigen. So rather than inefficient signals, like maybe, for instance, intolerance or an allergy, you're dealing with an antigen that never goes away. And that antigen that's constantly inflammatory. And this drives a kind of alternative differentiation fate. Cancer cells do the same thing because they avoid immune destruction. And you can characterize these exhausted T cells. It's a progressive lineage that they differentiate over time after repeated exposure, and they lose polyfunctionality, that is multiple cytokines per cell, self-renewal capacity, cytotoxicity, and ultimately they are characterized by co-inhibitory molecule expression, as well as the expression of lineage supporting transcription factors that I've listed here. Again, it's important to understand here that at least in the way that we talk about them now, exhausted T cells are a lineage. It's a differentiation state with an epigenetic signature that prevents them from really changing inherently. And so we'd seen these metabolic features. So the question is, does metabolic insufficiency characterize exhaustion, or does metabolic insufficiency cause exhaustion? And that's what we really wanted to take on, with the understanding that when T cells enter tumors, they get metabolic stress right away, but exhaustion takes time to set in. So we asked this question from the perspective of hypoxia, which is a common metabolic stress that is experienced by T cells. So here's that same L-shaped pattern I showed you before. When T cells infiltrate tumors, they differentiate down this lineage, and these are the most terminally differentiated and hypofunctional cells. And indeed, if you use this kind of terminology, your ability to synthesize IL-2 goes down. They express this transcription factor tox and multiple co-inhibitory receptors. So what we did is said, what if we look at them from that metabolic lens, right? And so we infused these animals with pimenidazole. That's that hypoxia tracer I showed you in that first slide. And if we stain for pimenidazole, what you can see is that hypoxia exposure is not a uniform feature of tumor infiltrating T cells, but rather exhausted T cells seemingly kind of uniquely are severely hypoxic. And if you don't like this pimenidazole stain, here's some HIF-1 alpha staining to go along with it. So this told us hypoxia must play a role in T cell exhaustion. So to get at this, we really wanted to explore this, but hypoxia has really been explored in T cell differentiation before, but it's kind of been tenuous. A lot of the work has been done in genetic systems by deleting, for instance, HIF-1 alpha or its upstream regulator VHL. And interestingly, hypoxia exposure alone can drive co-inhibitory molecule expression and effector function. And in fact, if you grow therapeutic T cells with just IL-2 and hypoxia, it actually improves their in vivo activity. But I'm a signaling guy by training. And so hypoxia is not the only signal that exhausted T cells receive. And like I told you before in the last slide, a well-known driver of exhaustion is persistent antigen. So we hypothesized that hypoxia may alter the way that T cells respond to other signals and vice versa. So to do this, we developed an in vitro system where we could explore this by taking ova-specific T cells, so the splenocytes. We activated them in vitro. We didn't want to explore this role in activation, but rather the later stages of differentiation. So we activated them overnight, and then we put them into different conditions where we made them alone, just added IL-2. We co-cultured them next to B16 melanoma cells, or next to B16 melanoma cells that were expressing the cognate antigen, and thus promoting chronic stimulation. And we did that in and out of a hypoxia chamber. And then we did that for 5 days, and then we looked at the cells. And what we found, really interestingly, was that T cells could withstand being next to tumor cells. They could withstand being in hypoxia. But when you started playing with the antigen, things got different. When T cells experience chronic stimulation, they turned on a little bit of PD-1, a little bit of TIM-3. But if they experience both at the same time, chronic antigen and hypoxia, they turned on a lot of PD-1, most of them turned on TIM-3, but far more importantly, they lost their effector function when we restimulated them in vitro. So in other words, experience both at the same time drove the cells to be in a different fate than any of the things individually. Now, this is a very messy experiment because the tumor cells change when you put them in hypoxia as well. So we broke down the system and recapitulated the continuous activation phenotype by using something that wasn't alive, rather stimulatory magnetic beads. These are off-the-shelf beads you can buy from Life Technologies. And we activated the T cells overnight and then put them into 4 conditions. We either expanded them, continuously added beads, so they continuously got stimulated, and we did that in and out of a hypoxia chamber, and we completely recapitulated the phenotype. So again, stimulation, continuous stimulation by itself drove a little bit of PD-1, a shift in TIM3 expression. Hypoxia alone, you kind of got this middling phenotype, but experiencing both at the same time drove the majority of the cells to be PD-1 high, TIM3 positive. They carry many other features of exhaustion, including multiple co-inhibitory receptors, the transcription factor tox. But again, all of these things are just correlative. You have to ask their function. So when you re-stimulate those cells, it's really only the cells that experience hypoxia and continuous stimulation at the same time that drive the exhausted phenotype. And indeed, we're even able to recapitulate that exposure to hypoxia alone actually drives a superior effector phenotype. Now, were we just acutely poisoning the cells, or did we actually change their differentiation? So to do this, we repeated the experiments and then removed the cells and put them into just IL-2 with no stimulation and in fact, not even any hypoxia. And we do that, actually the phenotypes diverge even more. If the cells were continuously stimulated, they became alone in normoxic conditions, they became phenomenal effector T cells. But if they experienced hypoxia during that time, they were very poorly polyfunctional. Even if we rescued the oxygen tension, we were not able to rescue these cells. So what were we doing to these cells? So we did RNA sequencing, and I hope what you can appreciate here by this PCA plot is that experiencing continuous stimulation plus hypoxia drove a unique transcriptional program that was different from either signal alone. And that was really driven by this clusters 1 and cluster 7, which is interesting, a bunch of interferon stimulated genes. Now, interestingly, if we ran gene set enrichment analysis compared to previously published transcriptomes on less differentiated progenitor cells versus terminally exhausted T cells, we were really producing cells that very much resembled terminally exhausted T cells. Now, I want to be completely transparent. These cells did not look like TIL. We were not capturing all the other features of the tumor microenvironment, but the things that drove them to be the most different from the other 3 populations looked more like exhaustion. What our RNA-seq did confirm was a pattern of BLIMP-dependent transcriptional repression driving down that same transcription factor we talked about before, PGC1 alpha. So we went back to this PGC1 alpha thing. What is PGC1 alpha actually doing? So to do this, we returned to our PGC1 alpha overexpression experiments using GP100 specific T cells that would respond to melanoma. And we put them back into melanoma-bearing mice. But rather than read them out functionally, we sequenced those TIL to understand what were we doing when we overexpressed this gene. So here's the gene ontology pathway, where to tell you what the pathways were that were being upregulated. Now, the first one on this list is graft versus host disease, which is a bunch of effector cytokines, because remember I told you these T cells work better. But the number 2 gene on this list is the regulation of reactive oxygen species. Kind of an unappreciated feature of metabolic reprogramming is that when you make new mitochondria, you have to kind of quench the reactive oxygen species that are going to come from those new mitochondria. And indeed, if we measured the mitochondrial reactive oxygen species using mitosocs in T cells that were overexpressing PGC1 alpha, we saw very significant repression, which suggested that maybe ROS is also playing a role here. And indeed, consistent with data from several other groups, including Peter Siska, Jeff Rathmel, Jess Thaxton, you can see that at the steady state, terminally exhausted T cells in tumors harbored elevated levels of mitochondrial ROS. And indeed, if we stain for mitochondrial ROS in our in vitro exhausted system, the continuous stimulation plus hypoxia group had the highest levels of steady state mitochondrial ROS, which made us wonder, is mitochondrial ROS sufficient to drive exhaustion? So to do this, we actually utilized, we had to kind of go back to undergraduate biochemistry and understand where this ROS is coming from. Well, hypoxia induces ROS because the electron transport chain that we all memorized in our undergraduate years is running inefficiently. And so it generates ROS at complex 1. Now we can mimic this in the absence of hypoxia by using a complex 3 inhibitor called antimycin A. It inhibits complex 3, which causes ROS to build up in the first 2 sets of complexes. And indeed, we could culture T cells with no chronic activation, no hypoxia, but just in low doses of antimycin A and generate mitochondrial ROS. But interestingly, we could add another inhibitor, rotenone, which inhibits complex 1, and actually bring that mitochondrial ROS back to baseline. And this was a pretty interesting set of experiments, because if we cultured T cells in antimycin A, we actually drove an exhausted-like phenotype, PD-1 high, TIM3 positive, but more importantly, those cells failed to function. But what was really, really cool about this experiment is that if we added another inhibitor and reduced the ROS back to baseline, we actually rescued the T cells. So this is telling us the T cells would rather have no mitochondrial activity than dysfunctional mitochondrial activity. So we kind of had it wrong. It wasn't the loss of mitochondrial activity, but the presence of dysfunctional mitochondria and the production of ROS that induced a dysfunctional state. So could we kind of get around this by using antioxidants? So we used N-acetylcysteine, which is a very common cell-permeant antioxidant. That was enough to reduce antimycin A-induced dysfunction, the red to the purple. It was also sufficient to rescue chronic stem plus hypoxia-induced dysfunction, the dark red to the light red. Now, we can't do this experiment in vivo because N-acetylcysteine would get eaten up by the tumor cell, but what we did instead was we did genetically using overexpression of an antioxidant protein called GPX1, which is a target of PGC1 alpha. You can see GPX1 overexpression reduced ROS in tumor infiltrating T cells, and it improved their T cell effector function. So in the interest of time, I'm going to skip over this slide and really go after how we can therapeutically modulate ROS, because we really can't remove chronic antigen, right? We want those T cells to seed their antigen in tumors. But could we remove hypoxia? And to do this, we could use drugs that would target the tumor's metabolism uniquely. This is one thing that we've explored, and I would encourage you to reach out to look at this published work from Cancer Immunology Research using the type 2 diabetes drug metformin. Metformin specifically targets tumor cells' metabolism and lowers their oxygen consumption, and that reduces the hypoxia in the tumor really dramatically, and it synergizes very, very well with anti-PD-1. We can also reduce hypoxia by correcting the vasculature using VEGF inhibition. This is the tyrosine kinase inhibitor, axitinib. And I hope what you can appreciate here, the low dose of axitinib normalizes the vasculature, reduces hypoxia, and that also synergizes very well with checkpoint blockade immunotherapy. So all this is to say is that the persistent antigen that the T cells receive drives this kind of transcriptional program that includes repression of metabolic reprogramming. And that creates a vulnerability so that when cells enter into hypoxic regions, they become sensitive and start producing ROS. Now that ROS changes the way that the T cell signals, including changing the metabolism of the cell, but also reinforcing pathologic signaling. And we think this is the main driver of T cell exhaustion in our state. Now, I'd like to change gears here and really talk about another feature of the tumor microenvironments. And this is something that really kind of drove the generation of my laboratory, which is that while we always talk about how nutrient deprivation, hypoxia in the tumor can inhibit T cell function, some cells really do quite well there. Is that the tumor microenvironment is not miserable for everyone. In fact, some cells thrive. And it's always just kind of boggled me. And so I really wanted to explore this in more detail. So what we've shown previously is that the more metabolically active the tumor cell, the more dysfunctional like the T cells that were inside. But this is not the case for every T cell. Some T cells thrive in this environment, and the one that we really explore a lot is the regulatory T cell. Regulatory T cells are a key feature of tumor microenvironments, and they're highly overrepresented. Here's the data over here, is that normally in the lymph nodes, they're a very small proportion of CD4 positive T cells, but in the tumor, they're more than half of the CD4 positive T cells are regulatory. And they're not just inert bystanders. If you look at those Treg cells and we look at their cell cycle progression, The vast majority of them are in cell cycle, and a significant proportion of them are in S phase. So they're not just sitting there idle, they're actually actively proliferating, which means whatever metabolic feature they have, there allows them to thrive. So we started exploring the metabolism of these cells, the regulatory T cells in the tumor microenvironment, starting first by understanding how Treg cell function was linked to the metabolism of the environment. Now, again, we've previously shown that tumors with higher metabolic activity carry a more dysfunctional infiltrate. So does that hold up for Tregs? So what we did is we purified Treg cells from various tumor microenvironments of different metabolic activity. Here, this is just their glucose consumption. And what we found, these are 3 different lines, that if we isolate Treg cells from each of these environments, the more metabolically active the tumor, the more functional the Treg, right? This was the exact opposite of any other T cell. That if you were to do this experiment and look at gamma interferon production from T cells, it would be the opposite, right? So this is telling us that Treg cells have a very, very different relationship with metabolites compared to conventional T cells, especially glucose. And indeed, this is a plot of that I showed you in the first couple slides. If you take conventional T cells and you activate them in vitro, they immediately change their glucose metabolism. But Treg cells are loath to engage in glucose metabolism, which you can see tabulated here. And indeed, if you pulse them with a glucose tracer, if you bathe them, in fact, in a glucose tracer, they really are very reticent to even take up glucose, which tells us that this is a different part of Treg cell biology. And indeed, this has been shown before that expression of FOXP3 actually represses the ability to take up glucose. Now, it's not 100%. There is actually a proportion of Treg cells that do take up glucose. So we asked, like, is this actually a relevant, does this tell us something about their function? So what we did is we took tumor-bearing regulatory FOXP3 reporter mice and pulsed them with the glucose tracer. And here's, again, those data. The conventional T cells love this stuff. The Treg cells are very reticent to take up this tracer. Then what we did is we sorted them based on their glucose uptake and asked them to suppress in a suppression assay. Again, this is where we co-culture them with dye-labeled conventional T cells. And so you can see if we don't have any Tregs, the cells divide. If we don't stimulate them, so those are the maximum minima of the assay. And here are the data, super exciting. If the Treg cells were avid for glucose, they were actually very, very poor suppressors. Whereas the Treg cells didn't take up glucose, they were actually phenomenal suppressors. So this is, again, very, very different than conventional effector T cells. So we did RNA-seq on these 2 different populations, and what we found is that while the glucose avid Treg cells, the ones that took up the tracer, they were still Tregs, they still expressed FOXP3. They had this kind of overall weaker Treg cell signature. They did not express a lot of the canonical Treg kind of stability genes like neuropilin, Helios, the CD25, and so on. We assessed their long-term stability by putting them into immunodeficient animals, and we added conventional T cells to see how well they would suppress those conventional T cells. And you can see here that they ultimately lost FOXP3 at 7 weeks post-transfer, and they were unable to control the colitis that was produced by these naive T cells in this immunodeficient environment. And in fact, even culturing Treg cells in high glucose conditions, in 25 millimolar glucose, was enough to actually make them suppress less. So this was really exciting. This told us that glucose and Treg cells, these things did not go really well together, and this may help them survive in a glucose-poor environment, like the tumor. But could we actually understand what they were eating if they by looking at these cells? And what we found, interestingly, is that the cells that were not avid for glucose, right, these NBDG low Treg cells, they actually still expressed all of the glycolytic genes. So this is really interesting. They weren't taking up glucose, they weren't doing glucose metabolism, but they were still expressing all of the genes. Now, what we found is that the cells that were not avid for glucose, these regulatory T cells, they actually overexpressed the terminal steps of glycolysis, LDH A and B, and the monocarboxylate transporter SLC16A1. These are transporters that move short chain sources of carbon, monocarboxylates, across the cell membrane. And the most abundant monocarboxylate that's present in tumors is lactic acid. Okay, so lactic acid is the byproduct of tumor cell metabolism that eats glucose and ferments it into lactic acid, the so-called Warburg effect. So is this what these Treg cells were eating? So we just started playing around. Lactic acid has long been known to be immunosuppressive. And indeed, if you take conventional T cells and you start titrating in lactic acid, they stop proliferating. This is just a proliferation die. But if you do the same experiment with regulatory T cells and you titrate in lactic acid, they actually not only tolerate it, they thrive in this condition. And they also maintain their suppressor phenotype when they're expressed, when they're cultured in lactic acid. We did metabolomics to determine what these Treg cells were doing with lactic acid. You can see here when we compared them to conventional T cells in blue, the green regulatory T cells, they did take up lactates, they converted it to pyruvates, and it entered the TCA cycle. But far more importantly, they weren't just burning lactic acid, they actually exported the lactate-derived carbon into the cytosol and formed higher order intermediates like PEP and glucose 6-phosphate. So in other words, you may remember this from your biochemistry courses, these Treg cells were actually doing gluconeogenesis. They're actually generating higher order intermediates, which is really important for creating nucleotides and non-essential amino acids that you need for proliferation. Indeed, if we used an inhibitor of gluconeogenesis called 3MP, we were actually able to inhibit the proliferation of regulatory T cells in tumors. So what were the critical components of lactate uptake by Treg cells in vivo? All this stuff was in vitro. So to do this, we obtained a floxed allele from Jeff Rothstein at Hopkins, who studies this lactate shuttles in neurons. And so he created a floxed allele for MCT1, and we bred it to FOXP3 to delete it in the Treg cells. Now, importantly, these mice don't get sick. So the Treg cells don't need this transporter to stay alive or to suppress kind of homeostatic immunity. However, when we implant these mice with tumors that would produce a lactate-rich environment, the Treg cells fail and they result in slow, indolent tumor growth. If we look inside the tumor microenvironment of these animals, you can see that the T cells, the conventional T cells, the CD8s and CD4s, they're more proliferative and they produce more cytokine. So what happens to the Treg cells in these environments? Well, remember, I told you at the beginning, when Treg cells enter tumors, they repress glucose metabolism to stay alive and eat lactate. But if they can't eat lactate, they start to take up glucose. They start to try to take up glucose. When this happens, kind of all the other features fall in place. They can't proliferate. They become less stable. They start to express PD-1. And when you pull them out of the tumors and ask them to suppress, they no longer carry their suppressor capacity. So finally, could we use this in a therapeutic system? So what we did is we bred our allele to the inducible FOXP3 specific Cre, and we were able to recapitulate the phenotype in 3 different models when we acutely interrupt lactate production using tamoxifen treatment. And again, we were able to see that slow indolent tumor growth. But when we do that in combination with anti-PD-1, we're able to mediate tumor aggressions in about 30% of mice. So this told us that lactic acid metabolism represents this kind of supplemental metabolic pathway that Treg cells use to maintain functional identity in these kind of inflamed, metabolically distinct tissues. Now, finally, for the remainder of the talk, I want to talk about a different environment. This is unpublished work from our group, but one that I think is really exciting. So we're going to go a little bit long, but I think it'll be fun to talk about. And that is really a completely different type of environment, which is making therapeutic T cells. So these are things you've read about before: CAR T cells, TCR engineered T cells, and expanded TIL from biopsies. All of these things result in is when you grow T cells in vitro and then reinfuse them back into patients. And we've been really interested in understanding how these T cells interact in these environments when they get infused back into the tissue and how they infiltrate into hypometabolic conditions. And so we've been really focused on providing an edge for T cells to function in these unique environments. But we've been kind of ignoring a different kind of environment, which is this environment, the environment in which we make the T cells, which is not hypometabolic, but hypermetabolic. The typical media that people use to make T cells for therapy, called ex vivo 15, has nearly 40 millimolar glucose, which is about tenfold more than the serum glucose of most healthy people walking around. So is this a stressor that T cells experience when you expand them? And is that part of why T cells fail when we infuse them into patients? So what we wanted to do to really explore this is really understand whether or not T cells can actually, what the metabolic differences are when you activate T cells in vitro compared to your typical response in vivo. So to do this, we took OT1 T cells and we either activated them with our cognate peptide in vitro and expanded them like like most of us normally do, or we transferred them into mice that were bearing an ova-expressing virus, an acute viral infection. And the reason for this is that if you do this T cell, if you make these T cells, they proliferate like crazy, become effector T cells, and then promptly die. Whereas if you do this experiment, these T cells proliferate like crazy, become good effector T cells, but then form long-lived memory, the kind of T cells that we want in cancer therapy. So whatever the metabolic differences are here are something that we can explore. Now, intriguingly, at the effector time points, these T cells are very similar looking functionally. They produce cytokines, they have the same transcription factor profile. But what if we look at them from that metabolic lens? And so while it was very different to capture functional differences, their metabolism was very, very distinct. That is, T cells that were expanded in vivo carried higher mitochondrial mass, higher ability to store lipids, higher increased respiratory capacity, that feature of long-lived cells. Now, interestingly, these T cells, despite the fact that we're feeding them glucose like crazy in vitro, they were no different in their ability to take up this glucose tracer. However, how they handled glucose was different. The thing that the in vitro T cells did compared to the in vivo T cells did was that they were fermenting that glucose into lactate. That thing that I was been telling you about for the whole talk. We've known about it since the 70s, that when you activate T cells, they ferment glucose into lactates, but that these T cells were doing it at the steady state while we were growing them, and that wasn't happening in vivo. So how do we make T cells for therapy? We stimulate them with CD3, we co-stimulate them with CD28, we give them boatloads of IL-2. And as it turns out, if we measure glycolysis in these cells, as we give them these different things, IL-2 can do it a little bit, CD28 does really nothing on its own, CD3 starts the process, CD28 enhances it, and IL-2 drives it even more. So in other words, all of the signals that we're giving to T cells tell them to ferment glucose into lactic acid. And that this is something that is evolutionarily conserved, but maybe for making T cells for therapy, it's maybe not the greatest thing that we should be doing for cells. Now, it turns out we've been exploring this a lot as a lab previously. Glucose is, of course, really important for cells, not just for energy, which I think is where we get kind of caught up, but it's also, of course, a key synthetic intermediate. It's how you make nucleotides. It's how you stabilize antioxidants, and it's how you make amino acids. Now, one thought would be, could you just limit glucose metabolism while you grow them? Could you grow them in serum lactate conditions? And indeed, you can improve ACT. This was shown beautifully by Nick Restifo and Luca Gattinoni back in 2013. But if you lower glucose, because you need it for nucleotides, you actually severely limit the ability to grow T cells, which is a key feature of adoptive cell therapy. So we've been studying these metabolic shifts in T cells, and what we found is that glucose metabolism isn't just regulated by the expression of these glycolytic enzymes, but also by gatekeeper enzymes. And one of the biggest gatekeeper enzymes is this giant hollow enzyme called pyruvate dehydrogenase, which converts pyruvate into acetyl-CoA for use in the TCA cycle. And it is post-translationally regulated by PDHK1, its kinase. And this kinase, we found a few years ago, can be phosphorylated through different T cell stimulatory pathways. TCR, CD28, and IL-2. Here you can see its phosphorylation almost immediately upon T cell stimulation on tyrosine 243. Now, this is very good for acute effector functions. These T cells, these signals turn off glucose oxidation, promotes glucose fermentation, and that's good for these acute kind of inflammatory effector functions for T cells. Now, you can inhibit this kinase using a small molecule called dichloroacetate. It was previously used in to treat lactic acidosis, and indeed, you can see when you activate T cells, they start fermenting glucose. When you add DCA, it goes back down to baseline. So DCA is a way to reroute glucose into the mitochondria rather than kind of stopping glucose metabolism wholesale by lowering or inhibiting glucose. And when you do this, you actually have no impact on the expansion of T cells because you're rerouting glucose into the mitochondria. All right, so could we temper aerobic glycolysis, just the end products, during the expansion process and promote a more in vivo-like metabolic state? So we activated GP100 specific T cells and expanded them with IL-2. And we just added DCA while we made the cells to prevent them from doing that aerobic glycolysis. And when we do this, we get this more in vivo-like metabolism. The T cells have higher mitochondrial mass and higher respiratory capacity. Again, that value of long-lived T cells. So then what we said is, could we use them as a therapy? So we expanded those T cells, and then we treated mice bearing 3 millimeter B16 melanoma tumors. Now, importantly, we didn't lymphodeplete the mice. We didn't give them any vaccination in vivo. We didn't treat the mice with anything, just the cells. And when we do this, we get a beautiful therapeutic index. So you can see the control T cells, they barely work. This is very similar data from many groups. But if we expand the T cells with dichloroacetates, we get a lot of tumor clearances, but more importantly, all mice receive benefit from these cells. This translates into humans. We can take PBMCs and grow them with DCA. We can provide them with the effector phenotype. We can even do this from TIL. If we take melanoma patient TIL from biopsies and expand them with lots of IL-2 plus or minus DCA, we can also provide that same respiratory capacity to cells that have already been in the trenches and already exhausted. So this is super, super exciting. We also explored whether or not we could do this with CAR T cells. So we activated human peripheral blood T cells. We transduced them with a CAR. We expanded them. Again, you can see that same respiratory capacity value, but now we could use those CAR T cells to treat mice that were bearing antigen expressing tumors. And indeed, you can see we can provide a survival benefit to those animals. So finally, I'll just finish with some of the mechanistic studies which are not finished yet, which is, how does redirecting glucose flux actually affect that T cell function in vivo? What were we doing that caused this dramatic phenotype? What I'll tell you is, we were cancer immunologists. So immediately we said it must be that we were improving the T cell function in the tumor microenvironment, that the T cells were getting into the tumor and they were working better there. So we went right to the tumor. And we were really disappointed. We found that the DCA expanded T cells had no changes. They still got exhausted. They still lost their mitochondrial mass. And they had the same effector function as the control T cells, even though the mice that were treated with the control T cells would die, and the mice that were treated with the same amount of DCA expanded T cells would live. So what was happening here? So I asked Andrew, who was working on this, the student working on this project, to mix them, to do a competitive experiment, and maybe we could compare the two to one another. And this is what ultimately led to the answer. We used congenically mismatched OT1 T cells, so Thy1.1 for homozygotes versus Thy1.1, 1.2 heterozygotes, and we transfer them in 50-50, right? And it didn't matter how we did the assay; we could flip-flop these just for importance. So this is the mix before we put them in, and then we put them into mice that were bearing tumors. And what we found was that there was a numerical advantage. That if we looked at the tumor 5 days later, the DCA expanded T cells were at a much greater proportion than the untreated T cells. But that actually, even a day after we infused them, that they were already at a numerical advantage. And more importantly, this numerical advantage had persisted remarkably over time. That 4 months later, long after the tumor was gone, or after in the control, after we resected the tumor, those T cells that we put in were a dominant proportion of the circulating immune cells. So nearly 6% of the immune cells that were constantly circulating in the mouse were the ones that we put in. So we all know this. We all do adoptive transfer. You put in 1 million T cells and you expect to find maybe 100,000. And we think that maybe the metabolic stress of expansion is playing a role in that. So finally, to conclude this part of the talk, hyperphysiologic stimulation combined with hyperphysiologic glucose drives this kind of phenotype that we know is evolutionarily conserved in T cells, aerobic glycolysis. But that is maybe not what we want to do for a long period of time while we grow T cells in vitro. Rerouting glucose to the mitochondria via targeting of PDHK1 maintains mitochondrial capacity and health without affecting expansion or potentially transduction efficiency. This metabolic health translates actually into increased longevity, which means a numerical advantage in vivo and ultimately superior therapeutic efficacy. And we've shown that in both mouse and human systems. And our data suggests that this kind of sustained aerobic glycolysis severely hinders the ability of T cells to kind of re-enter into the tumor, into the immune system in general. And that ultimately affects their ability to kind of become and establish and engraft and be part of the immune system. So finally, just to kind of put this all together, I talked to you today about providing metabolic support to tumor infiltrating T cells, reprogramming the tumor, understanding how there are suppressive metabolic relationships. What I want you guys to take away from all of this is that when you're thinking about stimulating the immune system, whether or not that's to a virus, to tumors, to fungal infections, anything, even to self, the fact is we have to be thinking about how these T cells metabolically support that function. And that's something that's very, very critical to understand as basic immunologists and as translational immunologists. Finally, I'd like to thank the people who did the work. This is my microenvironment. The first part of the talk was driven by Nicole Sharping. The second part of the talk on Treg cells was driven by Mac Watson. And the third part was driven by Andrew Frisch, Yang Wang, and Dinos Lantos. Thank you for your attention. Happy to take questions.
Thank you so much, Greg, for an excellent talk. We are sort of a little over time, but I think if for those who can stay, we can go on for another 10 minutes or so. Greg, if you can stay also to answer
happy to.
questions. Great. So I'm going to take advantage of being moderator to ask the first question, and that is sort of wearing my infectious disease hat. So obviously, tumors are a specific microenvironment with nutrient depletion, but they're also very particular microenvironments in some infectious diseases like TB. So for example, the TB granuloma, where it's this conglomeration of immune cells that infiltrate to sort of box in the bug, and is also known to be hypoxic. So is it a stretch to think that this type of microenvironment may also influence T cell function in this setting?
Yeah, absolutely. What we're finding now is that, you can find exhausted T cells in a lot of places when you start really looking. And now that we've kind of, come up with, if you will, the appropriate kind of, kind of parameters in which we measure T cell exhaustion, we're finding them everywhere. And absolutely, you can find them in granulomas. You can find them in the kidneys of lupus nephritis patients. You can find them in type 1 diabetic islets. A lot of places, when they're places with a lot of damage and where you would expect persistent antigen to be around, you start to see exhausted T cells. So absolutely the case, but definitely in granulomas, that's something that's really interesting.
Thank you. So we'll take some specific and then some general questions. So here's a question. The tumors treated with PGC1 alpha modified T cells started to grow, but regressed around day 30. Can you speculate on why this happened so late?
Yeah, this is a great question, and this is one that I always tell people that are in, that work for me in the CAR T cell space, is at least in the CAR T cell world, you start to see this growth happen first, and everybody thinks the experiment failed. But that's actually, we think, the infiltration of T cells into the tumor and the in situ proliferation. So they're actually kind of, it's almost like pseudo progression. The tumors grow and they immediately start to shrink. And this is something that's seen in a number of different kind of gene modified CAR T cell approaches.
Thank you. How important is antigen density and TCR affinity and avidity in exhaustion? So if one were to compare peripheral blood T cells versus those infiltrating. Yeah,
this is a great, I think all of these features play a role. So whether or not it's avidity is very important, affinity is very important. Chronicity, of course, is extremely important as well. So these are data that have been explored by various people over time. Andreas Scheidinger just recently had a very nice paper in JXMed looking at chronicity as well as antigen avidity, just using Synfecal as the model antigen to really explore what were the features that drove exhaustion. And all of these features all kind of play a role and all kind of converge upon the exhaustion phenotype.
Thank you. So there are other suppressor cells like myeloid-derived suppressor cells, there are other T cell subsets, Th17s. Do these infiltrate the tumors and also get exhausted?
Yeah, this is a great question. So lots of suppressor phenotypes, lots of suppressor cells present there, or cells that act like suppressor cells. All those things have been observed. Interestingly, I would argue that their cells behave exhausted because they become exhausted because they have the same kind of metabolism. So intriguingly, MDSCs, tumor-associated macrophages, have also been shown to take up lactic acid. That's by work by Ruslan Medzitov, a few other groups as well. So we think that the metabolism of cells kind of is part and parcel, if you will, with their ultimate immune function. But it's a great question. It's one that I think we need to continue to explore about how the fuel and the function are linked together.
Here's another one. What might be other ways to reroute glucose to mitochondria other than the PDHC1 alpha? Could it be some amino acid or other metabolite?
Yeah, I mean, there's a lot of different ways that you can do this. There are. I mean, PDHK1 is kind of a pretty well-known way to do this. You'd still have to, I think, you'd still have to temper that axis to get there. You could also, for instance, inhibit LDH, and this was shown by Warren Leonard's group at the NIH. You could inhibit LDH and also get glucose into the mitochondria. There are other ways that you could explore this too. Toying with the mitochondrial pyruvate carrier, that's another way that you could explore. Getting more glucose into the mitochondria. Deleting HIF1 alpha, that would be another way to do this. So there's a couple of different ways you could explore this.
Thanks. Here's someone asking about the procedure of isolation of the TILs in vitro. So how does the isolation procedure and the sorting impact the metabolic readout of these TILs? So different types of enzymatic or mechanical digestions and so on.
Yeah, I think this is something that, this is a tough, kind of cautionary tale, if you will, right? I mean, whenever metabolism is something that's very, it's very current, right? It's something that's happening now, right? So, when you're looking at some of these features, especially things like glycolysis, which we know can happen immediately, right? Can change immediately when the cells get the right signals. So you have to kind of, interpret your data with caution, but there's not a lot that you can do about it, right? I mean, if you're gonna study TILs, you have to study them as soon as you can, and you have to isolate them the best that you can. Now, what I can say is that we have explored different ways to kind of isolate T cells from tissues, whether or not that's mechanical digestion, whether or not that's typical proteases that are used like collagenase or cold proteases, cold active proteases. These things can provide some alterations, but the key thing is just simply treating your cells the same way, right? So if you're going to compare them, either compare them within the same compartment, like TIL to TIL, or comparing them or treating, for instance, your circulating T cells with the same kind of cocktail, and at least understanding that there will be changes induced by the isolation procedure, but it's either that or don't do the experiment at all. And so until we come up with better ways to do this, this is going to be part of the confounder in the process.
Yeah,
I know that's a very conceptually unsatisfying answer, but it's unfortunately what we have to deal with.
So I'm not sure I get this completely, but maybe this will make more sense to you. So could the TIL metabolic change in non-responding patients be actually a result of older, naive T cells in some individuals that have already lost their mitochondria prior to the cancer?
Yeah, this is a great question. So the features of exhaustion and senescence are the same features as that, not the same features, but have very similar features to what occurs during aging, kind of. And while the oxidative theory of aging has come and go and come back and come, it's like a pendulum of whether or not the oxidative theory of aging is true. What is very kind of unequivocal is that T cells from older individuals are less metabolically sufficient. They have poorer mitochondria, and yes, it could absolutely be the case. Now, we actually adjusted for that variable in our analysis. Age was not a variable. There were differences in age, but we could still see the differences in the severity of T cell loss. That was still a defining and significant feature in our data set.
Thank you. So here's a participant who says, we've recently come to appreciate that metabolites also act as signaling molecules. So wanted to ask, what about oncometabolites other than lactate and their effects on T cell fate?
Yeah, this is a big thing, right? There's all kinds of metabolites that are enriched in tumor microenvironments. They could be just elevated metabolites. They could be oncometabolites. We're exploring many of these. There are the kind of canonical ones, like lactate is one of these. 2-hydroxyglutarate is a very exciting one. There's been increases in there are things like kynurenine. These are things that we know that are highly elevated in tumor microenvironments that play a role in T cell function. We think it's a major things that we haven't been looking at. We've been really, I think, as immunologists, exploring what's missing in the tumor microenvironments. But I think what we're learning is that it's really what's present there that's really truly immunomodulatory. And I think there's a lot to study in that space. Stay tuned from work from our lab. A little early to talk about it, but we've been really interested in understanding some of these interactions.
Great. I think we'll have one last question, and that is, what about the possible consequences of enhancing these CAR T cells via the metabolic control after complete eradication of the tumor? So if these cells remain in the host for longer, is there any benefit or pathology to those enhanced cells?
Yeah, that's a great question, and it's one that I think I hope is kind of what we're getting. I mean, we're using cancer as a model right now, so to speak. It's not that I don't care about it, learning all these cancer, treating cancer better, but I think we're learning a lot about the immune system in general. And I think that what we're taking away from this is that there is most certainly features of this that are going to be important. Now, these enhanced CAR T cells, we know they live longer. We do not think they become pathogenic over time. We do not think that they ever transform. But yeah, the question really is could you use the same ideas to make better memory T cells in response to vaccination, to make better memory T cells for other types of diseases that you might treat with cellular therapy? CAR T cells are now being considered in Parkinson's and cardiovascular disease. These are things that in CAR Treg cells for autoimmune diseases. These are things that we're absolutely considering, and I think are going to be the next era, if you will, of immune metabolism research.
Excellent. On that note, Greg, so those are all the questions that I can see. So we're going to have to wrap up now. We're getting to 4:10. So thank you so much for your talk. I know you've indicated that you can provide your email address, and for those who have questions, they could email you their specific questions.
Yeah, feel free to follow me on Twitter or DM me on Twitter or email me directly. I'm happy to answer any questions that I didn't get to.
Fantastic. And there are lots of compliments in the chat box for you. So thank you so much and for sharing your really elegant experiments and data and unpublished data with us.
Very much appreciate it. And thank you again for the invitation. Take care, everyone.
Thank you. And thank you to IUIS and Lena for organizing this, to Thermo Fisher Scientific for the support, and of course to the audience for your attention and your questions. That wraps up today's session.
Bye-bye.